Plants turn sunlight into sugar and provide chemical energy for most living organisms on the planet, including various microbes. Through millions of years of evolution, all plants have evolved immune systems to protect themselves against the vast majority of microbial invaders, including viruses, bacteria, fungi, and fungus-like oomycetes. However, aggressive pathogens can cause serious diseases on their host plants. One of the most common and important plant diseases is powdery mildew. This disease can be found in over 10,000 plant species, including many agriculturally important crops such as wheat, barley, cucumber, grape, strawberry, and rose. Powdery mildew is caused by ascomycete fungi that belong to the order of Erysipeles. These fungi are obligate biotrophic pathogens that require living hosts to complete their life cycle. Therefore, they steal nutrients from host plants to keep them alive. After landing on the surface of a host plant, a fungal spore germinates in a few hours, and then develops an opressorium by which the fungus penetrates the plant cell wall, and further differentiates a feeding structure called the hostorium. Establishing a functional hostorium in the host cell is essential for fungus to establish a mycelial network, capable of producing asexual new spores from conidiophores in about 4 to 5 days. In about a week, massive fungal mycelial and conidiophores form an extensive network in the surface of the host tissue. In 10 to 12 days, a layer of whitish powder-like mildew gets accumulated in susceptible host plants. That's why this disease is called powdery mildew. It is important to point out that the hostorium is the only fungal structure in the hostile host cells, and essential for the success of the fungus. Conceivably, the hostorium is the Achilles heel of the fungus, because it is the only reachable target of the host defense. Based on earlier microscopic studies, the hostorium is separated from the host cell cytoplasm by an interficial membrane named the extrahostorial membrane, EHM for short. The EHM represents the host pathogen interface and the critical background for both the host and the fungus. However, the origin and the biogenesis of EHM and the molecular warfare between the host and the pathogen in this background is largely unknown. The Zhao Laboratory at University of Maryland characterized a broad-spectrum powdery mildew resistance gene called RPW8 from the model flowering plant Arabidopsis. Plants containing the RPW8 gene locus are resistant to all infectious powdery mildew, whereas plants lacking RPW8 are susceptible. Interestingly, although novel in deduced protein structure, RPW8 utilizes a conserved immune signaling pathway to activate resistance. A key question about RPW8 is how it activates broad-spectrum resistance via the same pathway of race-specific and basal resistance. Strikingly, by analyzing the subcellular localization of a biologically functional RPW8-YFP fusion protein, it was found that the RPW8 protein is only made in the host cells invaded by powdery mildew, and specifically targeted to the surface of the fungal feeding structure, the hostorium. A single optical section of a confocal image of the isolated hostorium from host cells expressing RPW8-YFP indicates that this protein is precisely localized at the extrahostorial membrane, i.e. the EHM. RPW8 is the first protein ever identified in this mysterious yet important interficial membrane. As demonstrated by this short movie, RPW8-YFP is targeted to the EHM by membrane vesicles. Small green dots carrying the RPW8-YFP are moving, decking, and then fusing into the EHM. This is a reconstructed 3D confocal image showing that the fungal opressorium penetrated the host cell wall and formed a hostorium inside the host cell. The hostorium is encased by the EHM, where RPW8 is specifically localized. Further studies showed that RPW8 somehow enhances the formation of a callose-enriched cell wall-like structure encasing the hostorium and triggers production and accumulation of hydrogen peroxide, H2O2, in the interface. These defenses are highly hostorium targeted. Thus, in terms of a defense action, RPW8 functions like a molecular missile. This finding also explains well why RPW8 confers broad-spectrum resistance to powdery mildew. Simply put, the RPW8 missile targets the feeding structure of all powdery mildew pathogens. Conceivably, the RPW8 protein must contain a guidance signal for the EHM-specific localization. Indeed, mutational analyses identified two regions from the same basic residue-enriched motif, R, KR, KXR, K, to be essential for the EHM targeting. Thus, this short motif may represent the zip code of the EHM. Disruption of these two targeting signals can abolish the EHM targeting of the RPW8 and its mediated resistance to powdery mildew. The identification of RPW8 as a molecular missile for antifungal immunity in plants opens up several exciting new questions for future research. How does RPW8 activate defense at the interface? What's the trafficking queue from the EHM that directs RPW8 vesicles to the EHM? How does the EHM originate? Is it from invagination of the host plasma membrane or de novo synthesis? Do host cells engage an EHM-specific membrane or protein trafficking pathway for defense? If so, what are the trafficking components involved? Finally, can one use RPW8 as a delivery vehicle to target any antimicrobial protein to the host pathogen, thereby engineering novel, Haustorium-targeted resistance in crop plants? This research in the Zhao Lab has been supported by the National Science Foundation, USDA, NIFA, and the Gates Foundation.